This is chapter 2 of IB biology book, the proteins. So, first of all, we're going to talk how proteins or the polypeptides synthesize and broken down. So, condensation. Proteins are made of amino acids, and the structure is there's a central carbon and attached to a carboxyl group on the left or in either side, and then there's amino group on the other side. And in the central, it's one H and then the R functional group. So condensation happens while the OH from the carboxyl group and the H from the amino group combine to form water and draw out from the two amino acids. So in reverse, it's the reaction that when you're adding water into it and it breaks down the polypeptides to amino acids. So it's hydrolysis. And just be aware that there are 20 different amino acids synthesized on ribosomes, and after they're synthesized, they can be modified to form more derivatives of the amino acids. And 9 of the amino acids are considered essential amino acids for humans, and there's one that's conditional. It's required for younger children, but not for adults. And then most organisms use the same 20 amino acids with the genetic code, with few exceptions. Then the 4 levels of structure. There are primary structure, secondary structure, tertiary structure, and a quaternary. So from the first one, basically, it's just the sequence and the chains of the polypeptide. The bond that's responsible to binding amino acids together is called peptide bond, and which is a covalent bond. Primary structure is very important because they are responsible for all the later structure. Because amino acids, when they are assembled on ribosomes, they form chains immediate after the amino acids are binding together. So a slight change in the primary structure will affect its conformation later and ability to function. And there's no protein functional at this level, but it's just extremely important. So secondary structure is the 2D, two-dimensional shape, the alpha helix and the beta sheets. So the hydrogen bonds in the peptide chains are responsible for this level of structure. And the same thing, there's no proteins function at this level, but they're heavily involved in the tertiary level. So tertiary level, the final 3D structure of protein, and it contains the secondary structure. The reaction a lot, like ionic bonds, hydrogen bonds in the secondary structure, hydrophilic, hydrophobic reactions, and then there is disulfuric bonds between two sulfurs. And then the example of tertiary structure proteins is lysosome and the quaternary. So basically quaternary is just a protein or polypeptides that's a tertiary structure that interact with each other. So 2 or more polypeptide chains combine with each other. So it's the interaction between 2 different polypeptides, or is it called the subunits of the protein. And most likely the quaternary structure will increase its efficiency or its strength in a way, and examples are collagen and hemoglobin. Okay. So just to mention tertiary structure, how many bonds or interactions that's responsible for it. So like the pictures show here, the first one is disulfide bonds. The sulfur in cysteine amino acids will interact with each other, and that's they form a kind of strong bond. And then there's ionic bond between the groups. And then hydrogen bonds is basically the ones that actually formed already in secondary structure that's contained in tertiary structure. and there's hydrophobic and hydrophilic interactions. These are especially important to determine the 3D shape of the function, whether it's fibrous or globular, because in fibrous tertiary structure of proteins, they have high content of hydrophobic functional groups, so they result in a string long narrow shape, and which also gives this property of insolubility. And globular is when the hydrophobic groups that actually centered towards its center, surrounded by its hydrophilic part, so which is more sensitive to change, and then it's soluble, most likely soluble in water. Then proteins and the polypeptides. So you just have to understand that proteins can mean one single polypeptide or a combination of several polypeptides. And then these are the examples how polypeptide can function on its own, but most likely a lot of functional proteins they actually combine with each other and form quaternary structure of proteins. So examples of collagen and hemoglobin. For collagen, when they're combined, they have like they give it more tensile strength, and for hemoglobin, it just increases its efficiency to transport oxygen. Then functions of proteins. So we all know that proteins have multiple functions, and then these are just examples. They're not all class of proteins. There are more functions of proteins, but I think these are all we need to know. So there's structural support, muscle movement, transport, storage, hormone, enzyme protection. So here I just give a more specific function of specific proteins if you need to know, but I think these are good enough. Oh, sorry. I think these are good enough if you just memorize them. So, okay. So, denaturation and renaturation of proteins. Most likely, if a protein is denatured, it will not return to its native conformation. And the causes of denaturation are heat and extreme pH. So, how heat change or break the protein's shape is by causing vibrations within the weak bond, because tertiary structure of protein, the bonds are weak, so it's very subjective to the changing temperature and the pH. So when it causes vibration, it breaks the bonds in its quaternary, tertiary, and the secondary structure. But be aware that the peptide bonds within the sequence of the polypeptide will not be broke by this process. However, since it loses its shape, it will not be functional. And for pH, it's more like when it's not within the range of optimal pH or within the range of this pH tolerance, then it will change the charges on the R group, thus break the ionic bonds, and then change the overall conformation of the protein. So just be aware that renaturation can happen, just happens very rarely, and most likely we say that denaturation is permanent. Okay. So protein mix. So unlike genome is one individual's total genes that holds is fixed, the proteome is all of the proteins produced by a cell, tissue, or organism, and it's very unique, subject to the individual or the single cell, and it can vary among time. So for this, you just need to know that proteome actually is larger than the human's genome, and since genes contains all the information that we need to make the protein, however, our genes can be turned on and turned off. Sometimes we choose not to express certain genes, and then And then I just know that protein is very unique. Every individual has a unique protein, and then it will change according to the activities your cell is doing at the moment. So protein sometimes give us what the the perspective of the status of the cell at the moment, not predicting it. I hope that makes sense. So yeah, thank you for watching and good luck on your exam.